Good afternoon and welcome to Chimeric Therapeutics presentation. Chimeric is developing groundbreaking CAR T cell therapies for solid tumours based on scientific research conducted by leading US CAR T experts at the City of Hope Cancer Center in Los Angeles. The technology known as CLTX-CAR T uses chlorotoxin, a peptide discovered in scorpion venom, as a tumour-targeting component of the CAR to reprogram T cells. This technology is currently undergoing phase 1 clinical trials at City of Hope. Today we have Dr. Christine Brown, the scientific founder, and executive chairman Paul Hopper. Paul is the founder of Chimeric. He has more than 25 years of experience in the biotech and healthcare and life science sectors. Focused on startup and rapid growth companies, he's served as founder, chairman, non-exec director or CEO for more than 14 companies in the US, Australia and Asia, including Viralytics, Imugene, Prescient Therapeutics, Polynoma and Suda Medical. His experience covers extensive fundraising in US, Australia, Asia and Europe, and he has experience in corporate governance, risk and strategy. Dr. Brown is a faculty member in the departments of hematology and hematopoietic, nice pronunciation, cell transplantation and immuno-oncology, and one of leading CAR T scientists in the US today. As Deputy Director of the T-Cell Therapeutics Research Laboratory, Brown, the Heritage Provider Network Professor in Immunotherapy, provides scientific oversight for the preclinical research program, as well as the ongoing clinical trial program focused on the development of CAR-engineered T cells for the treatment of solid tumours. I'll now hand it over to the guys, and just as always, any questions that do come through, please feel free to submit via the Q&A box down the bottom. I'll now hand it over to Paul and Christine. Thanks very much.
Look, thank you, Simon, and thank you everyone for logging in to hear the presentation this afternoon. And I just give a welcome to Professor Christine Brown, who's dialing in from California, and I think it's about 6:30 in the evening. So Christine is going to help me out on the deeper science matters that may arise. So if we could just start off here on the executive summary. Chimeric Therapeutics is an Australian company that was incorporated to license technology from City of Hope, which was discovered and developed at Christine's lab at City of Hope. So we have the global exclusive rights to the CAR T therapy, which is the subject of this presentation today. Our first drug that we're targeting with the CAR T is a CAR T for glioblastoma, or what you would call adult brain cancer. And the therapy is known as chlorotoxin CAR T therapy, and you'll see the acronym CLTX, which is chlorotoxin. The unusual part about what we're doing here is that the CAR T actually has its basis, or part of its construct, coming from the Deathstalker scorpion. It's toxin, it's poison, and that's the 36 amino acid peptide. So it's a rather unusual sort of background to a CAR T, but it is quite unique, and Christine will talk about that in a little while. We're targeting glioblastoma. It's one of the deadliest cancers, and the outlook for patients with that disease is not great. We've demonstrated, or should I say, the lab has demonstrated that the chlorotoxin, that is the scorpion component of this, can be successfully built into a CAR T cell, and we'll talk about that a little bit later on. At the moment, CAR T therapy has demonstrated some incredible success in blood cancers, and I think it's fair to say that it's one of the hottest places in immunotherapy or oncology research in the world today. And Big Pharma is watching it very, very carefully, and certainly ploughing significant resources into it. We go to the next, Simon, please. So this is a novel platform technology which has applications across many cancers, not just GBM, and they would be maybe prostate, melanoma, lung, etc. We are already in clinical trials, so a phase 1 clinical trial at City of Hope is underway. So that's the culmination of 5 to 10 years work under Christine and her collaborators at City of Hope, all the preclinical work, extensive tox and safety work, and we got the IND approved by the FDA at the end of last year, and 2 patients now on the study, and we're recruiting further patients. So we expect to have some data out in the first quarter of next year. The trial's being run at City of Hope, which is one of the leading cancer centers in the US, named the 11th Beth Cancer Center Hospital recently. So the strategy after the phase 1, if we get positive signals out of the phase 1 study, would be to go to the FDA and seek approval to do a phase 2 pivotal study. That is a study that could be, if it demonstrates what we believe the drug can do, that could be allowable for an approval effectively. And we'll talk about that in a little while. We have an extraordinary senior management team. Unfortunately, I'm not able to reveal who they are just at the moment because they're finishing up, but they come out of 2 of the most successful CAR T companies globally. We have a great board. Leslie Chong, who's also on the board of Imugene, is a glioblastoma expert. She came out of Genentech, where she ran a number of their brain cancer trials, and Leslie Russell, who is a cell therapy specialist as well. As you probably know, in biotech, you don't actually have any real hard assets. The asset here, as in most biotechs, is the intellectual property, and we have very robust IP. There are no CAR Ts anywhere that use chlorotoxin, that is the scorpion poison, as part of the construct. So we're pretty comfortable with that. Manufacturing is done. We have GMP manufacturing at City of Hope, which is an acknowledged and highly regarded CAR T manufacturing facility. And the drug, we believe, will be safe, and it's had extensive prior experience in humans as an imaging agent. So we believe that the safety side of it is going to be okay, unlike a lot of the blood cancer CAR Ts, where you often get, or you can get, cytokine storm, which is a nasty negative response of the immune system. We've done a lot of work to get us into man, and maybe I could just ask Christine to just talk briefly about the comprehensive work in vitro and in vivo that was done to get us to the point where the FDA allowed us to go into a phase 1.
Great, thank you, Paul. So this work started over 5 years ago and really centered around evaluating patient human brain tumors for the chlorotoxin binding, and developing mouse models of brain tumors to evaluate the therapy. And what's important is that this therapy showed potent activity against brain tumors, and also exhibited no toxicity that was observed in these preclinical models. So we spent a lot of time optimizing the therapy, developing it further, and establishing a clinically relevant manufacturing platform to bring this to clinic. And this is what we'll talk to you about today.
Thanks, Christine. And just finally, we have a rather major differentiator to the blood cancer CAR Ts. This drug is given as an outpatient and does not require the hospitalization that the other CAR Ts require, which adds a huge cost to the therapy. And obviously that's a major concern to the reimbursement authorities and the insurers. Simon, let's go to page 5, which is technology highlights. And Christine, could I ask you to just run through a couple of these, the highlights on these, if you would, please?
Great. So I just want to reiterate some of the key highlights to this therapy. So really what we're talking about, that this is a novel immunotherapy that reprograms the immune system through developing this CAR T cell therapy to recognize solid tumors. And so one of the key aspects is that we believe that this therapeutic could be broadly applicable to many different solid tumors. And there's strong evidence based on our preclinical data and based on this agent also being a chlorotoxin peptide already being dosed into patients in other clinical trials. So there's strong evidence to support the safety of this therapeutic. And then in our lead indication, glioblastoma, we show that this therapeutic broadly recognizes brain tumor cells and many different patient tumors, many different glioblastomas, and they're highly effective at eliminating brain tumor models in mice. And then finally, the differentiator that City of Hope has a really robust translational infrastructure, as does my laboratory. So we've been able to take this from bench to clinic in just about 5 years. And so the clinical trial is underway, and we've started treating patients.
Thanks, Christine. If we could go to slide 6, please, Simon. I'm going to just explain briefly in layman's terms, which may seem a bit presumptuous given that Christine is on the phone, but what is a CAR T cell? So basically what they do is they draw blood from the patient, then by a process called leukapheresis, they spin out the T cells. And you may know that the T cell is one of the important arms of the immune system, but the T cell doesn't always pick up cancer cells. So what they do is they essentially reprogram the software in the CAR T cell. And if you look on the diagram here, you'll see the gray circle that says CLTX-CAR T cell. So that's after the T cell has been reprogrammed and becomes effectively a guided missile specifically against a target on the surface of the cancer cell. And in this case, it's a molecule called MMP2 and an associated complex. And this CAR T cell that we have here called CLTX-CAR T is very specifically directed to molecules on brain cancer cells and also would be applicable in other cancers. So that's effectively what it is. It's a reprogramming of a T cell and turning it into a very specific targeting cell against a precise target on the cancer cell surface. I hope that was okay scientifically, Christine.
I'm impressed. Great job.
Thank you. Can we go, Simon, to 7, please? You've had an introduction to Christine from Simon at the start, and her collaborator and fellow inventor is Dr. Michael Barish. So we're extremely fortunate to have both these people with us. The company has a 3+2 year contract research agreement with them, which basically gives Chimeric access to the lab and the people and the facilities and equipment within City of Hope. Next one, Simon, slide 8. Christine, could I ask you to take this, why it's unique?
Sure. I think it's, I just want to touch base on some of the unique aspects of this therapy. This is the first CAR T cell to use the chlorotoxin peptide. And so it's a very novel receptor that's engineered into T cells to allow it to recognize and then hopefully destroy a broad range of cancers. And so this is an important aspect. If you're going to have an effective immunotherapy, you want to make sure that the T cells are reprogrammed to recognize a large number of the malignant cells to really debilitate the growth of the cancer. And so this therapy has been developed to achieve that. Another important point, which I mentioned before, that chlorotoxin as a peptide has been in clinical trials and been shown to be safe. So it's been used as a radiotherapy and delivered into the resection cavity of patients, so delivered locally into the brain, and that's been shown to be safe by others, not us, but as a peptide. And it's also been used as an imaging agent, so using the chlorotoxin peptide fused to a fluorescent molecule and given systemically, intravenously, so that a surgeon could more precisely resect the tumor, given its specificity of binding malignant cells. And that as well has been well tolerated in patients. So we're really building on this foundation and now using the immune system, and not just to take advantage of this unique binding property of this peptide. And then finally, what's unique is really the experience from my program, my laboratory, and City of Hope for translating this therapy. So We think it's really exciting that we've already initiated a phase 1 study and really moves us forward quickly in the therapeutic development of this platform.
Thank you. Slide 9, Simon, thank you. Christine, just if you could let us, the audience, know what other cancers are available or could be available.
So our lead candidate is really focused on glioblastoma, and we'll talk about that later and the reasons for that. But what we think is really enabling and exciting is the broad range of tumors that have been shown to bind the chlorotoxin peptide, which we believe that means that this therapeutic could be very advantageous in treating and potent in treating. That includes tumors such as melanoma, prostate cancer, small cell lung cancer, and other brain tumors.
Thank you. Could we go to 11, Simon? That's the media attention. Just quickly here, Christine's work was the subject of a major peer review article in Science, in the very prestigious Science Translational Medicine. It was the cover article. We had an enormous amount of response to it, and we are still receiving calls from people interested in the technology, willing to explore collaborations, etc. If we could go to the next one, Simon. If you have a look, just if you Google CAR Ts, you'll see that it is one of the hottest areas I mentioned. They talk about cures, they talk about the immunotherapy revolution. So it's an area, it's an area of research which is very, very keenly sought. And I expect we will see some further drugs the first drugs in solid tumors coming out in the not too distant future. Hopefully this one. If we could go to 13, there's just for those of you who are interested on the financial side, there's been some big deals. Back in 17, Gilead acquired Kite Pharma for 11 billion. In 18, Celgene acquired Juno for 9. This year on NASDAQ, a company called Legend Biotech did the biggest biotech IPO. They raised 424 million. They actually had bids into the book for $1 billion. And then just recently, a week or so ago, and this is quite a good comp to Chimeric, Innate Bio announced that they were going public and they're raising $86 million as part of that. If we could go now to 14, please, Simon, glioblastoma. Christine, you might just take us through that and mention some of the way it's treated today and the lack of new therapies.
Right, so glioblastoma is a primary brain tumor, meaning it arises in the brain. It's one of the most aggressive and common adult brain tumors. And I think what's critical, and one reason this is our lead indication, is that that there has been no real therapeutic breakthroughs in decades. So median overall survival remains at under 10%, closer to 5%, and that really hasn't changed in many decades. It's an orphan drug indication, and therapies are getting approved, such as bevacizumab, or what others might know as Avastin, on really limited improvements in overall survival. So Avastin was approved with not improving overall survival, and so there's a relatively low bar for getting therapies approved because there's really no other therapeutic options. And so we think we have a huge opportunity to make a significant impact in this patient population.
Thank you. Let's go to 15 moment.
So let me just to give a few points about our ongoing phase 1 study. This started this year. It's led by Dr. Venom, my clinical partner at City of Hope, who's chair of neurosurgery. And so this is a phase 1 safety feasibility study. Our goals are really to establish the maximum tolerated dose. We've treated 2 patients thus far, and things are going well. Patients are doing fine. We anticipate treating approximately 18 patients to establish the dose that we would move forward with in a phase 2 pivotal trial. And we can I think that's a good summary for at this point.
Thanks, Christine. 16, please, Simon. I won't repeat the phase 1, but if we see very positive signals out of the phase 1, we would go to the FDA and talk about a pivotal study, just as Avastin was approved on a phase 2 study for GBM. And that would be a 50 to 75 patient study across 4 to 6 centers and do the manufacturing outside. If we could go now, Simon, to the slide 18, because I think we've talked about the manufacturing. IP, as I mentioned, is very robust. No one else is using a chlorotoxin, a scorpion-derived peptide as in a CAR T, so we're very comfortable with that. Next slide, Simon. I've mentioned the board of directors, both people with relevant experience to the business. And the options open to the company going ahead. There's multiple ones. First would be the sale of the company. Second would be to develop it independently. And the third would be a strategic partnership with Big Pharma. I think if the phase 1 performs as we expect it will, then we're going to have lots of option in the early stages of this drug's life. Next one. This is just a little bit of a further to the earlier slides, illustrating how some of these preclinical, this is prior to phase 1, companies have sealed major deals, back in 14, Pfizer doing 80 million up front, and then Baxalta in 16, 105 up front, Gilead and so on. So big interest in anything that has a lot of promise. So we're very excited. And Simon, can you take me to the last slide? We've got a very busy year ahead next year. As I mentioned earlier on, the technology and the announcement that it's gone into Chimeric has generated a lot of interest. So we are looking at collaborations with other companies in complementary technologies. We have the sponsored research agreement with Christine's lab over the next 3 to 5 years, and we're going to build out the platform for other cancers such as melanoma, prostate, and breast. We think we'll probably do patient updates March, June, September, and December. And if the trial tracks according to what we hope it will do, we'll start getting ready for the phase 2 trial towards the end of next year, like finding the CRO to run the phase 2 and then find a manufacturing facility to make the phase 2 drug as well. So we'll have a busy year next year and lots of exciting things. So thank you everybody for your attention, and we'd be happy to answer any questions if there are any.
Great, thanks Paul, and thanks Christine. Just a reminder for those that are on the webinar, if you did want to ask a question, just do so via the Q&A box down the bottom. First question is, when do we expect phase 1 to complete and how many patients are recruited for phase 1?
Right, so we've got approximately 18 slated for the phase 1 trial. Recruitment rates obviously determine how fast you get the trial completed, but we've said 15 to 24 months, depending if later on in the trial we decide to bring another centre on in the States. But Christine, is that in accordance?
That's accurate with our expectations.
Great. Next question is, what has been the company's funding history to date, and are there any intentions to do further pre-IPO rounds or otherwise?
Right, so we have done a convertible note issue for about 4.3 million a couple of months ago, which has funded us nicely. We're considering what the opportunities are in terms of capital raising going forward, but at this stage we haven't yet made a decision.
Great, thanks Paul. What would the potential structure of a phase 2 trial look like?
Right, there was a little summary there, but again, Christine, I'll have a go at it and I hope I've got it right. We think probably 15 to 75 patients across 4 to 6 centres in the US. Anything you want to add to that, Christine?
I think that's what we're expecting. And, we've also discussed potential sites in Australia as well. So we're looking at all those as options.
Great, thanks. That concludes the Q&A. There's been, for a private company, there's been some serious interest in terms of attendance, which is great to see in terms of the prospective nature of the technology. Paul and Christine, I might hand back to you to provide closing remarks and anything else that you want to finish on.
Yes, well, look, I've been involved with a lot of different biotech companies, and when I met Christine and saw the work that she and Mike Barish had done, I was extremely excited and was very intent on being able to license it. So for me, it's a great privilege to work with scientists like that. And I think if you dive deep into the science and read the papers that are out there, I think you'll share my enthusiasm for the great promise that I think this technology has. So we're in for an exciting time.